Carbohydrate has bifunctional groups, which basically are hydroxyl groups and carbonyl groups. So it can be described as polyhydroxyaldehyde or polyhydroxyketone. Carbohydrate can be further classified into simple carbohydrate, including monosaccharide, oligosaccharide, and polysaccharide, and the complex carbohydrate, sometimes called glycoconjugate. Let's focus on monosaccharide, which is a basic unit or building block of all kinds of carbohydrates. Its chemical formula is CHO in the ratio of 1 to 2 to 1, when the least carbon is 3. Its name is usually ended with O's. We always call the general name of monosaccharide by mentioning the number of carbon and ended with O's. For example, triose is a monosaccharide containing 3 carbon atoms. As mentioned earlier, carbohydrate might be aldehyde called aldose or ketones called ketose. So the general name could be extended with aldo or keto in front of its name. For example, ketotriose means a monosaccharide containing 3 carbon atoms with ketone functional groups. Likewise, aldotriose means a monosaccharide containing 3 carbon atoms with aldehyde functional group. Let's practice which aldotretose, ketopentose, and aldohexose are. Let's move on how the number of carbon is allocated in monosaccharide molecules. The carbon number 1 is at the end of the molecules where the ketone or aldehyde group is. The other number is arranged as follow order, like these molecules. C1 is at the red arrow pointing. Likewise, this molecule is the same. Monosaccharide has variety of isomer due to the chiral properties of the carbon atoms, specifically called stereoisomer, including enantiomer, epimer, diastereomer, and anomer. Let's begin with enantiomer. Enantiomer occurs due to chiral carbon atom and both molecules, which are mirror image to each other. One form is assigned as D form, and the other is assigned as L form. To determine whether the structure of monosaccharide is D or L form, the last chiral carbon atom is configured. If the hydroxyl group is written on your right hand side is D form, while on your left hand side is L form. Can you determine which of the following structure is D or L form? In nature, D monosaccharide is much more dominant than L monosaccharide. Next, epimer is an isomer that only one position of hydroxyl group differ to each others. However, it must be in the same configuration and carbon number first of all. Here are examples: D-glucose and D-mannose is epimer to each other at carbon number 2, while D-glucose and D-galactose is epimer to each other at carbon number 4. Diastereomer, in case that it is not enantiomer and that it is not epimer, so it has become diastereomer. For example, D-mannose is a diastereomer with D-galactose. Let's practice which molecules are enantiomer, epimer, and diastereomer with D-ribose. Next, we are focusing on anomer. Anomer is an isomer in the ring configuration of monosaccharide. When the linear structure of monosaccharide is dissolved in water, the molecule saccharides into a ring structure using a lone pair of electrons in oxygen and the last chiral carbon atom to share with the carbon group of carbonyl group to form a hexagonal ring called pyrene ring and or pentagonal ring called furan ring. The previous carbonyl become an anomeric carbon atom, resulting in two isomers, namely alpha or beta, when the hydroxy group is written below or above the plane, respectively. The rest of the hydroxy group are written according to the fact that if it is on the right hand side of the linear structure, it will be written down the primary structure in vice versa. Let's practice to write this structure. Monosaccharide has their derivatives: acid sugar, alcohol sugar, amino sugar, deoxy sugar are examples of monosaccharide derivative. Acid sugar is derived from oxidation of monosaccharide, resulting in changing the last hydroxy group and or the carbonyl group to carboxylic acid. Glucuronic acid and galacturonic acid are samples of acid sugar derived from glucose and galactose respectively. Likewise, alcohol sugar is derived from reduction of monosaccharide resulting in changes the carbonyl group to hydroxy group. Glycerol, sorbitol, and xylitol are examples of alcohol sugar derived from glyceraldehyde, glucose, and xylose, respectively. Amino sugar is derived from the addition of amine or amide group into the sugar. For example, glucosamine and N-acetylglucosamine are derived from the addition of amine and amide group into glucose, respectively. The oxysugar is derived from the reaction to remove oxygen molecule from the monosaccharide such as deoxyribose found in DNA. The chemical reaction specific to monosaccharide is in reducing sugar due to the presence of carbonyl group or hydroxy group at anomeric carbon atom. To test for whether it is a reducing sugar, tolerance reagent or Benedict solution is used. Reducing sugar turns tolerant reagent to silver coated surface, while Benedict solution to change to cupric oxide precipitate. Both tests yield acid sugar. Here are the functional of certain monosaccharide where you have a look through. Next, we are focusing on oligosaccharide derived from at least two molecules of monosaccharide bound to each other with glycosidic bond. Those include disaccharide from only two units of monosaccharide such as maltose, lactose, fructose, thihalose, isomaltose, and cellobiose. And the oligosaccharide like galactose oligosaccharide called GOS and long-chain fructose oligosaccharide called LC-FOS. When an alpha-D glucose joins a beta-D glucose with a glycosidic bond by using the carbon number 1 of the alpha-D glucose and the carbon number 4 of the beta-D glucose, becoming a disaccharide called maltose. The specific glycosidic bond in maltose is called alpha-1,4 glycosidic bond. In this case, a maltose molecule exhibits 1N as a reducing end due to the freely presented carbon number 1 in the beta-D glucose subunit, and exhibit another N as a non-reducing end due to the freely present carbon number 4 in the alpha glucose subunit. The full name of maltose is recognized as alpha-D glucopyranosyl 1,4-glucopyranose or pyranosal. From non-reducing to reducing N. Remember, all monosaccharides are reducing sugar. What about disaccharide? This line indicates the structure of disaccharide. Can you guess which reducing or non-reducing sugar? Oligosaccharide comprise of at least 3 monosaccharide unit in the molecule joining together with glycosidic bond. For example, raffinose, an oligosaccharide composed of glucose, galactose, and fructose, mainly found in nuts and beans. Maltotriose, an oligosaccharide composing of 3 units of glucose, mainly found after digestion of starch. Moreover, certain oligosaccharides are found to be beneficial to gut microflora and supplemented in baby milk for improvement of baby development. Let's move on polysaccharide. Polysaccharide is made of many units of monosaccharide joining with glycosidic bond. The monosaccharide unit may be only one type of monosaccharide called homopolysaccharide, while at least 2 different types of monosaccharide called heteropolysaccharide. Let's begin with homopolysaccharide. Starch, a homopolysaccharide found as a food storage in plant, not found in animal. Specific alpha-1,4 and alpha-1,6 glycosidic bond joining among glucose subunit molecules. There are 2 subtypes of homopolysaccharide in starch. Those are amylose, of which a linear unbranched structure of polysaccharide, and only alpha-1,4 glycosidic bond are found. Another one, amylopectin, configure a branching structure of polysaccharide, and alpha-1,4 as well as alpha-1,6 glycosidic bond are found. Alpha-1,6 is found only at the branching point. Unlike starch, glycogen is found as a storage of energy source in animal body only. Like amylopectin, glycogen shows higher degree of branching than amylopectin does. It is found that the branching point is found even 8 to 10 units of glucose units. Glycogen stored in liver is aimed for maintenance of blood glucose, while it is an energy source during muscle contraction. Cellulose is another homopolysaccharide found in plant cell wall. It comprises of beta-D glucose joining together with beta-1,4 glycosidic bonds. Humans do not produce cellulose nor digest it for energy, so it acts as dietary fiber. Chitin is another homopolysaccharide found in fungi cell wall and exoskeleton of arthropods. It comprises of N-acetylglucosamine joining together with beta-1,4 glycosidic bonds. Like cellulose, humans do not produce chitin nor digest it for energy, so it acts as a dietary fiber as well. Heteropolysaccharide comprises of different types of monosaccharide. Glycosaminoglycans or GAGs or mucopolysaccharide is a major type of heteropolysaccharide of which disaccharide subunits are acid sugar and amino sugar. There are 4 types of well-known GAGs. Those are hyaluronic acid, composed of glucuronic acid and N-acetylglucosamine. Chondroitin sulfate, composed of glucuronic acid and N-acetylgalactosamine sulfate, or N-acetylgalactosamine. Carotene sulfate, composed of galactose and sulfated N-acetylglucosamine, and heparin, composed of hyaluronic acid and sulfated N-acetylglucosamine. glucosamine are examples. The second class of carbohydrate is called complex carbohydrate, known as glycoconjugate. It is clear that it is a carbohydrate forming complex molecule with other biomolecule classes. Proteoglycan and glycoprotein is a complex between proteins and carbohydrate, in which carbohydrate is the major and is the minor part in the complex, respectively. Glycolipid is a complex between lipids and carbohydrates.